Good morning, students. In our first session, we studied about what is genetics, definition, and about Gregor John Mendel and his studies and his history and his works on genetics. So, in this second session, we are going to deal about the first model organism in genetics, that is God and Peace, by some certain 7 characters studied by Mendel. And what's the reason for the white color in pea plant, white color flower in pea plant, and what are the terminology related to Mendelism and Mendelian inheritance, Mendel's laws of heredity, that means the law of dominance and the law of segregation. It is otherwise known as a law of purity of gametes. And also we are going to elaborately deal about monohybrid cross. Monohybrid cross. Finally, we are going to deal about the Mendel's analytical and empirical approach. Mendel's analytical and empirical approach. Okay. Now we are going to see about the 7 pairs of contrasting characters. 7 pairs of contrasting characters studied by Mendel. Okay, first one, stem length. The dominant triad is tall, the recessive triad is dwarf. Second, pod shape. Pod means fruit. The dominant triad is inflated and recessive triad is constricted. Inflated in Tamil it is known as Vingiya alladu Uppiya, whereas constricted in Tamil it is known as Irukkamutra. And third character, seed shape. The seed shape, the dominant character is round and recessive character is wrinkled. And fourth one, seed color. Seed color, dominant character is yellow and recessive character is green. Fifth character, flower position of the flower. Dominant character is axial and recessive character is terminal. Axial means leaf arise in the axil of leaves. Flowers arise in the axil of leaves. Terminal means flowers arise at the tip of the shoot. Here flower position, dominant character is axial and recessive character is terminal. Okay. Then flower color. Dominant character is purple and recessive character is white. Dominant character is purple and recessive character is. Then the last character, pod color. Pod means fruit. Green is the dominant character and yellow is the recessive character. Green is the dominant character and yellow is the recessive character. Now we are going to deal about what is dominant and what is recessive. The character which is expressed in F1 generation or during hybridization experiment, the character which is expressed in F1 generation is known as dominant character, and the suppressed or not expressed is known as recessive character. For example, when we crossed the stem, tall stem with dwarf stem in F1 generation, we can get all plants are tall. All plants are tall. Next we are going to see about 7 characters of Pisum sativum with genes. 7 characters of Pisum sativum with genes. For example, plant height. Plant height, the responsible gene is LG. Seed safe, the responsible gene is known as R. And cotyledon color, the responsible gene is known as I. A flower color, the responsible gene is known as A. Pod color, the responsible gene is known as GP. The responsible gene for pod color is known as GP. The responsible gene for pod form is known as V, and the responsible gene for flower position is known as FA. These are all very important in the form of 1 mark questions. Plant height it is known as LE. For seed shape R, cotyledon color I, and flower color A, and pod color GP, and pod form V, and flower position FA. In figure 2.4, you can see the Mendel's 7 characters in garden peas are shown on the plant's 7 chromosome. Okay, you can see the flower color and the seed color in one chromosome, pod shapes and flower positions in one chromosome, and pod color, pod shape, plant height and flower positions on one chromosome, pod color on one chromosome, seed shape on one chromosome. Next, can you identify Mendel's gene for regulating white color in peas? Let us find the molecular answer to understand the gene function. Now the genetic mystery of Mendel's white flower is solved. It is quite fascinating. Fascinating means vidhyalaya. Fascinating to trace the Mendel's gene. In 2010, the gene responsible for regulating flower color in peas were identified by an international team of researchers. So the gene responsible for regulating color in pea plants were identified by the international team of researchers. It was called a P gene A, which encodes a protein that functions as a transcription factor, which is responsible for the production of anthocyanin pigment. Here, anthocyanin is a pigment which is responsible for purple color. Here there is one gene called P gene A, which encodes a protein. That protein can function as a transcription, it can transfer the factor which is responsible for the production of anthocyanin pigment. So the flowers are purple. The flower with the anthocyanin pigments will exhibit its purple color. Pea plants with white flowers do not have anthocyanin. Pea plant with white flower means it don't have anthocyanin pigment, even though they have the gene that encodes the enzymes involved in anthocyanin synthesis. They also have the gene that encodes the anthocyanin synthesis. Okay. Researchers derived normal copies of gene A into the cells of the petals of white flowers by the gene gun method. Researchers delivered normal copies of gene A into the cells of petals, not sepals, petals, the colorful parts. That means white flowers by gene gun method. A gene gun method is the method in which the foreign particles, with the help of it, the foreign particles can send it into plant cells. There are so many methods are available for gene transfer, that is electroporation and gene gun method. Here they used gene gun method when a gene A entered in a small percentage of cells of white flower. When gene A entered in a small percentage of cells of white flowers, it is expressed in those particular cells and accumulated anthocyanin pigments and become purple. In white flowers, the gene A sequence showed a single nucleotide change that makes the transcription factor inactive. They find the reason. In white flower, the gene A sequence showed, in the gene A sequence, it shows a single nucleotide change that makes the transcription factor inactive. So the change can makes the transcription factor inactive. So the mutant form of gene A do not accumulate anthocyanin and hence they are white. Here the mutant and mutation. The sudden heritable change in the genetic material is known as mutation. The sudden heritable changes takes place in the genetic material is known as mutation. Mutation is a process. Okay. What about mutant? The organism which undergoes mutation is known as mutant. The organism which undergoes mutation is known as mutant. So, the mutant form of gene A do not accumulate anthocyanin and hence they are white. Mendel worked at the rules of inheritance and arrived at the correct mechanism. Before any knowledge of cellular mechanism, DNA, genes, chromosome becomes available. During the Mendel's period, there is no knowledge about the cellular mechanism, DNA, genes, chromosomal behavior, etc. So Mendel's insights and meticulous work into the mechanism of inheritance played an important role which lead to the development of improved crop varieties and a revolution in crop hybridization. So Mendel's mechanism of inheritance played an important role in the field of crop varieties and create a revolution in crop hybridization. Mendel died in 1884. In 1900, the work of Mendel's experiments were rediscovered by three biologists: Hugo de Vries of Holland, Hall Kohns of Germany, and Erich von Schirmack of Austria. Who the three scientists rediscovered the Mendel's work? Hugo de Vries of Holland, Karl Kohns of Germany, and Erich von Schirmack of Austria. These three scientists individually rediscovered Mendel's work. So next we are going to deal about the terminology related to Mendelism. Mendel noticed two different expression of a triad. Triad. Okay, example tall and dwarf. Triads are expressed in different ways. Triads, characteristics, due to the fact that a gene can exist in alternate forms. The gene can exist in two forms, that is alternate form, that is versions for the same triad is called alleles. Triads are expressed in different ways due to the fact that a single gene that is responsible for a character is existing alternate form or alternate version for the same character is known as alleles. If an if an individual has two identical alleles of a gene, two identical alleles, ஒரே மாதிரியான alleles, it is known as homozygous. That means capital T, capital G, homozygous tall. An individual with two different alleles, it is called heterozygous, capital T, small t. Mendel's non-true breeding plants are heterozygous. Mendel's non-true breeding. True breeding means continuously they are self-pollinated. So non-true breeding plants are heterozygous, two different. That is called as hybrids. That is called when the gene has two alleles. The dominant allele is symbolized with the capital letter and the recessive with small letter. When both alleles are recessive, the individual is called as homozygous recessive, small t, or dwarf plants. And individuals with two dominant alleles is called homozygous dominant, capital T, tall p plants. One dominant allele, one recessive allele, capital T, small t denotes non-true breeding tall pea plants. Non-true breeding tall pea plants, that is known as heterozygous. Next we are going to deal about Mendelian inheritance. Mendel's law of heredity. Mendel proposed two rules based on his observation on monohybrid cross. So based on his observation on monohybrid cross, he proposed two rules. Today these rules are called law of inheritance. These rules are known as law of inheritance. The first law is the law of dominance. And the second law is the law of segregation. These scientific laws plays an important role in the history of evolution. So Mendel proposed two rules based on his observation on monohybrid cross. These rules are nowadays called as laws of inheritance. The first law is known as the law of dominance, and the second law is known as the law of segregation. These scientific laws plays an important role in the history of evolution. So now we are going to deal about the first law, the law of dominance. The characters are controlled by discrete units called factors which occur in pairs. In a dissimilar pair of factor, one member of pair is dominant and the other is recessive. This law gives an explanation to the monohybrid cross, that is the expression of only one of the parental character in F1 generation and the expressions of both in the F2 generation. It also explains the proportion of 3:1 obtained at the F2. This law explains the expressions of only one parental character in F1 generation and the expression of both the parental character in F2 generation, and also explains the proportion of 3:1 ratio obtained at the F2. Now we are going to deal about the second law, the law of segregation or law of purity of gametes. So here alleles do not show any blending blending means mixing. Okay, both characters are seen as such in the F2 generation, although one of the character is not seen in the F1 generation. So here alleles do not show any blending. Both characters are seen as such in the F2. Both the characters are present in F2 generation, although one of the character is not seen in the F1 generation. During the formation of gametes, the factors or alleles of the pair separate and segregate from each other such that each gamete receives only one of the two factors. Only one of the two factors. A homozygous parent produces similar gametes. A homozygous parent produces similar gametes, and heterozygous parent produces two kinds of gametes, each having one allele with equal proportion. Each having one allele with equal proportions. Here gametes are never hybrid. Gametes are never hybrid. Students, next we are going to see about monohybrid cross. Monohybrid inheritance is the inheritance of a single character. That means only one character can be inherited. Here Mendel takes a plant height, tall and dwarf. It involves the inheritance of two alleles of a single gene. That means a single gene exists in two forms, that is tall and dwarf. When the F1 generation was selfed, Mendel noticed that 787 of 1064 F2 plants were tall, while 277 of 1064 were dwarf. The dwarf triad disappeared in the F1 generation only to reappear in the F2 generation. Here there are two terms, one is called genotype and second one is called phenotype. The genotype is the genetic constitution. What kind of genes are present in an individual or in an organism is known as a genotype. In simply it is known as a genetic constitution of an organism. Whereas the phenotype refers is an external structure, external characteristics. The term phenotype refers to the observable characteristics of an organism, an external structure of an organism. In a genetic cross, the genotype and phenotypes of offspring resulting from combining gametes during fertilization can be easily understood with the help of a diagram called Punnett squares, named after a British geneticist, Reynold Punnett. Once again, in a genetic cross, the genotypes and phenotypes of offspring resulting from combining gametes during fertilization can be easily understood with the help of a diagram called Punnett square. Okay, named after a British geneticist, Reynold C. Punnett. It is a graphical representation to calculate the probability of all possible genotype of offspring in a genetic cross. The law of dominance and the law of segregation give a suitable explanation to Mendel's monohybrid cross. The law of dominance and the law of segregation give suitable explanation to Mendel's monohybrid cross. Next, we are going to see about reciprocal cross, parimatra kalavi. In one experiment, the tall pea plants were pollinated with pollens from a two breeding dwarf plants. The result was all plants were tall. When the parent types were reversed, the pollen from a tall plant was used to pollinate a dwarf plant which gave only tall plant. The result was the same, all tall female versus a dwarf male, and a tall male versus dwarf female, matings are done in both ways, which are called reciprocal crosses. The result of the reciprocal crosses are the same. So, it was concluded that the triads, that is the characteristics is not sex dependent. The results of Mendel's monohybrid cases were not sex dependent. Now we are going to see about the monohybrid cross ratios. That is, genotypic ratio is 1:2:1 and phenotypic ratio is 12:1. Now you can see the parental generation, tall, true breeding, capital T, dwarf, true breeding, small t. In F1 generation, gamete formation takes place. In F1 generation, all plants were tall. Here you can see homogamous tall plant, heterogamous, one homogamous tall plant, two heterogamous tall plant, one homozygous dwarf plants. Okay, so genotype ratio 1:2:1. That means offsprings in F2, capital T 1, capital T small t 2, small t 1. So phenotypic ratio, tall is to versus dwarf, we can see phenotypic ratio is 3:1. Okay. The gene for plant height has two alleles, tall capital T versus dwarf small t. The phenotypic and genotypic analysis of the crosses has been shown by the checkerboard method or Folkline method. Now students, next we are going to see about Mendel's analytical and empirical approach. Mendel chose two contrasting triads, two different characters for each character. So it seemed logical that two distinct factors exist. In the F1, the recessive triad and its factors do not disappear and they are hidden or masked only to reappear in 1/4 of the F2 generation. He concluded that tall and dwarf alleles of F1 heterozygotes segregate randomly into gametes. Okay. Mendel got 3:1 ratio in F2 between the dominant and the recessive triad. He was the first scientist to use this type of quantitative analysis in a biological experiment. So Mendel was the first scientist to use the quantitative analysis in biological experiment. Okay. Mendel's data is concerned with the proportions of offspring. Mendel data is concerned with the proportions of Mendel's analytical approach is truly an outstanding scientific achievement. It's meticulous means kurmayana work and precisely executed breeding experiments proposed that discrete particulate units of heredity are present. Discrete தனித்து இயங்கக்கூடிய particulate units of heredity are present and they are transmitted from one generation to other. That particulate matter can transmitted from one generation to other. Now they are called as genes. Mendel's experiments were well planned to determine the relationship which govern heredity triads. This rationale is called an empirical approach. Laws that were arrived from an empirical approach is known as empirical laws. Okay students, in this second session we have studied about the 7 pairs of contrasting character and the gene which are responsible for the 7 pairs of contrasting character and their location on the chromosome, and which one is responsible for the purple color in pea plant, and what are the terminology which are used to or related to Mendelism and Mendelian inheritance, Mendel's law of heredity. We can see in the law of dominance and law of segregation. It is otherwise known as law of purity of gametes, and monohybrid cross. Monohybrid cross, inheritance of the only one character. Inheritance of only one character. Here the plant height, tall and dwarf. The phenotypic ratio and genotypic ratio of monohybrid cross. The genotypic ratio is 1:2:1 and the phenotypic ratio is 3:1. And finally, we see in Mendel's analytical and empirical approach. Okay, students. Now we can see what are the possible questions. What are the possible questions? Write the 7 pairs of contrasting characters. Write the 7 pairs of contrasting character. And for one mark, the 7 characters of physum sativum with the genes. The 7 pairs of contrasting characters can be asked in 3 mark questions. And who rediscovered the Mendel's work? Who rediscovered the Mendel's works? It may ask 2 mark or 3 marks. Then what's the meaning of alleles, and what is homozygous, and what is heterozygous, and what is hybrids? These are all 2 mark questions. Then write the law of dominance. 2 marks or 3 mark question. The law of segregation or law of purity of gametes. 2 mark or 3 mark questions. Then explain monohybrid cross. It's a 5 mark question. And here reciprocal cross, 2 mark or 3 mark question. Define reciprocal cross. What is genotype and what is phenotype? These are all 2 mark questions. And even certain time checkerboard for monohybrid cross will ask. Okay, students, we can see the third session. In third sessions, we are going to see about test cross, back cross, and dihybrid cross, etc. Thank you.